So we've looked at the overall organization of the innate immune system with various cellular, physical, and chemical defenses. Mostly we looked at phagocytosis and how that is controlled. We look at the adaptive system with antibodies and cytotoxic T cells, and we looked at the ways that T cells interact with other cells using T cell receptors, looking at proteins displayed on MHC. We looked at B cells, how those have to interact with some T cells. Now we're looking at things that can be triggered by either of those systems. So these are reactions that don't really fit as part of either system. So I definitely think of the complement system as an innate system. The reason I say that is because the individual parts of the complement system are identical between yours and mine. The complement system itself doesn't learn from infections, and it doesn't have any memory. It doesn't have any of the things that the adaptive immune system has. It's all just perfectly encoded by our DNA on the day we're born. So I really would put it with the innate immune system, but I also can see that it just isn't part of either system, or it's part of both. And that's because it can be activated by both systems. So the complement system is a group of proteins that are dissolved in blood. So they protect blood. And things they can accomplish. There are three major things the complement system can accomplish, and there are three major things that initiates the complement system's activities. So the things that it accomplishes, it can initiate inflammation. So it can trigger inflammation. It can opsonize pathogens. So the complement proteins, some of them act as powerful opsonins. So they make pathogens more attractive to phagocytes. And it can lyse some bacterial pathogens directly. So those are the three major things it can do. And it can be activated in three different ways. And so I want you to know these three things it can do and the three ways it can be activated. One way is called the alternative pathway. It's innate. Another is the mannan-binding lectin pathway, which is innate. And the adaptive one, the classical pathway, is where it's initiated by antibodies. So that's definitely adaptive. So this is a cool concept map showing the whole, showing most of the complement system. It is a complicated group of proteins that go through a series of cleavages, and they catalyze each other's cleavage in a complicated way. That, to me, is not that important. We can assume all these proteins are in the blood. All these proteins are in the blood, and they will do what they're supposed to do. So what I want you to know is the ways it can be triggered and the outcomes. And the way they've put this all in a concept map, again, is boxes have nouns in them. Arrows are verbs connecting these things. And so what they would have done is start with the outcomes and kind of move backward. Well, what triggers this? What triggers this? What triggers that? What triggers that? What triggers that? This is, again, a thing to do when you're studying something complicated, is build your own concept map. Connect nouns using the verbs that connect them. Okay, so let's get into how it's initiated. The innate pathways, they both involve some sensitivity one of the complement proteins has to a pathogen-associated molecular pattern. So certain PAMPs will directly trigger this whole thing by interacting with complement protein C3. So this protein will recognize things on the surface of a bacterium. In this one, this protein will recognize where mannose has bound to a pathogen. This can happen, fungi can often be like this. and mannose will bind to them in a specific way, and this protein will react once that happens. So these proteins are designed to react to very specific things. And that's really the level I want you to know it at. So the complement system can be triggered instantly by certain PAMPs on the surface of pathogenic cells and by binding of mannose to certain pathogenic cells. The complement system can also be triggered by an adaptive response. So anytime an antibody binds to anything, that antibody, its constant region, the region that is not specific to what it binds to, but the other part of it, that reacts. It changes its shape once the antibody has bound to what it's going to bind to. And so a lot of different parts of the body can sense whether that change in shape has happened, because that's a signal that the antibody has bound to something. And if an antibody binds to something, the body knows there are only a few reasons why that can happen, and they're all bad. So one reaction the body has to antibody binding to something is complement activation. So if there's something in the blood and an antibody binds to it, that's enough to trigger the complement cascade. So those are the three ways that the complement system can be activated. This is the adaptive or classical pathway. This is where the complement system was discovered as an extension of antibodies. So antibodies were thought to be able to kill bacteria, but they needed this other thing in blood to do it. So now we know antibodies don't directly kill anything. They bind to things, and they have all their effects through attaching themselves to stuff. But complement was seen as a complement to antibodies when it was first discovered, and that is part of how it works. So the complement system can do three things. It can initiate inflammatory responses, it can opsonize pathogens, and it can lyse some of them directly. And so this is showing the membrane attack complex, where after a series of reactions happens, if there is a bacterial cell membrane present, they will set up on that cell membrane, and these will form a pore. So this is a protein that has made a channel through the cell membrane, and this will basically break down the bacteria's ability to separate its cytoplasm from the outside, and that is death for a cell. So the complement system can do a lot of things that are useful for fighting off pathogens. Inflammation is another major topic in immunology. It is triggered in a lot of different ways, and it's involved in lots of different disease conditions, and it has effects we don't always understand. What we can say is that it is a non-specific defensive response. Something happens to the body, inflammation is part of limiting damage, it's part of healing, and it can be part of an immune response. And macrophages will usually take part in starting inflammation, but there are a lot of other factors involved. Inflammation has a lot of purposes. Things we want to happen because of inflammation are all of these different things. Inflammation can be involved in a lot of different aspects of an immune response, plus like a bigger thing it might be designed for is like helping us heal from injuries. And we could think of inflammation as a way of cranking up a local immune response, a way of making a lot of different cells more aggressive. And that's what we want to happen during local inflammation. What comes along with it is pain and tissue damage. And if the inflammation is excessive or if it's chronic, it can damage organ systems, and those systems can lose some function. So that's currently an area of research. It's like chronic inflammation in gut tissues leads to inflammatory bowel diseases. Inflammation in the central nervous system even low-grade inflammation can lead to a lot of problems. Excessive inflammation in muscle tissue or joints can lead to joint damage or slower muscle healing. So there's a lot of things that require inflammation to be controlled, and when it's controlled the wrong way, we know that causes health problems. So along with inflammation, fever is another non-specific, kind of crude way our immune response can fight pathogens. Fever is where we elevate our body temperature, and what it's trying to accomplish is making some of the anti-pathogen responses faster. And also it changes the temperature so that a lot of human pathogens won't be at their optimum. So fever itself is not the most powerful part of our immune system, but it is often a signal that our immune system is frantically ramping up and trying to defeat a fast-moving pathogen. So the last element of the immune system I want to talk about is natural killer cells. So natural killer cells are part of the innate immune system. They don't learn, they aren't random, they don't have like T cell receptors or B cell receptors. They have an innate function, but it is tied to adaptive functions. So we have to think back to MHC1. Abnormal cells should show their abnormalities through MHC1. If you go back and look, MHC1 is for displaying cytoplasmic proteins, whether they are normal proteins or evidence of a virus, displaying those and giving cytotoxic T cells a chance to look and potentially kill a cell. So that MHC1 system lets us identify infected cells and kill them and stop them from being pathogen factories. So pathogens have developed some ways around this. So some viruses will directly interfere with the system of assembling an MHC1 complex. C stands for complex, so MHC1. Other cells, like tumor cells, some of those will kind of accidentally stop making MHC1 because they're following the tumor gene expression program, not the cell's normal gene expression program. And if they do this, they escape from detection by cytotoxic T cells. So any cell that is not showing MHC1 is not being seen by the adaptive immune system. So those cells are abnormal. And so natural killer cells look for that in particular. They look for cells that are not displaying MHC1 and they kill those cells. They are part of innate immunity. So here's how we would think about it. The natural killer cell would have granules of damaging proteins, and it has one receptor that binds to another molecule on a typical cell, and binding with this pushes it towards attacking. It has another receptor that binds MHC1. And if it finds MHC1, that slows it down and stops it from attacking. So if both of these things are here, if the common molecule and MHC1 are both here, nothing will happen. But if no MHC1 is there to send that slowdown signal, then the natural killer cell will release these granules and kill this cell, potentially drilling holes in the membrane and potentially causing apoptosis at the same time. So natural killer cells are wild. They are part of innate immunity, and they exist because some pathogens can escape from adaptive immunity. And so they look for cells that know how to escape from adaptive immunity. And that it's just a brilliant brilliant depiction of the constant arms race between the host and pathogens. We already have two complicated, powerful layers of our immune systems, the innate and adaptive. And MHC1 is part of adaptive. Natural killer cells look for situations where that has been bypassed. Okay, so that's all the parts of the immune system I want to talk about. I have one more slide where I look at some classifications we see in the immune system. You'll see, when you're reading about the immune system, you'll see humoral immunity and cell-mediated immunity. You'll read about these if you read articles about developing vaccines, and about how the immune system controls COVID or influenza, or things like listeria, bacteria that are intracellular. Well, let's break it down. Humoral immunity is what can dissolve in blood and lymph. So antibodies and the complement system. And so they protect our blood and our lymph. The humoral immune response is what's finding exposed pathogens that are not inside our cells. They're not hiding. They're moving between our cells, in the blood or in the lymph. Cell-mediated immunity is cell-mediated immunity is specifically for pathogens that are hiding inside of our cells. So cytotoxic T cells, again, are going to look for any host cell that is infected with something and kill it. Type 1 helper T cells are going to look for macrophages that have found something bad, and they are going to tell the macrophages to be much more aggressive. And in both of those cases, it's looking at things that are inside these cells. Cell-mediated immunity could be seen as a way to find stealthy pathogens that are hidden inside cells. So viruses, for example, are often controlled by cell-mediated immunity. So I bring this up partly because if you look for resources for learning the immune system, you will hear oversimplifications. You'll hear these strange things that I've seen looking for videos that could be useful. And you'll hear YouTubers say this, and you'll hear some A&P professors say that like humoral immunity is antibacterial and cell-mediated immunity is antiviral. That is an oversimplification of both of those things. And I want you to think about this. So intracellular bacteria, those exist, and those have to be controlled by cell-mediated immunity. Virions have to travel in blood and lymph. They have to be controlled by humoral immunity. The flip side of this is people talking about COVID and do we have lasting immunity or don't we? And they're looking at antibodies. That's humoral immunity. And many of them are ignoring T cells. So they're ignoring cell-mediated immunity. And there's, for all we know, T cells might be the more important part of fighting off COVID. So don't let people make these oversimplifications when we know it's more complicated. And the main reason I bring this up is that we can't really predict what's going to be most important for a pathogen. Every new disease has a complicated way of interacting with the immune system, and we have to know what it is before we can think we know what it is. So this slide has a curiosity label on it. I wanted you to see it, but if you don't want to study it, don't worry about it. And then all the rest of the slides also have this label. So these are just screenshots from papers where people have tried to develop vaccines, and the situations are more complicated because the vaccine is giving them either a helper type 1 or helper type 2 response. And different diseases require different responses. So the helper type 1 response is going to lead us towards activating our macrophages, while the helper type 2 response would push towards aggressive antibody production. And so depending on what the pathogen is doing, we need one more than the other. But we don't have easy ways of telling a vaccine what to cause our immune systems to do. It's hard to push us towards one or the other, but it matters. So this is just an example of why it's challenging to make vaccines. There are some pathogens for which we just don't know how to make a vaccine that will work. We are lucky that viruses like influenza and the influenza virus and SARS-CoV-2, they're kind of straightforward, and our vaccines against them can be effective. But for some pathogens, we just don't have that. Last, I want to look at how vaccines work through herd immunity, and I'll come back to this when I talk about COVID in Unit 4. The idea with herd immunity is illustrated nicely by these colorful people. So let's just look real quick at what we have. Most people would be immunized in this case. So they've taken a vaccine or they've been exposed to something. And what we're looking at is the fraction of the people in this population who are immunized and healthy versus not immunized but still healthy. So these are the people who maybe didn't get the vaccine or the vaccine didn't work on them or whatever. So these are the people who could become infected. And then red, these are the people who are contagious. These are the people who could spread the infection. So in this scenario, we have 4 people who could potentially be infected, and only, let's see, I am not counting well. Only one of them gets infected. And the idea here is that in order for there to be an outbreak, there has to be a chain of transmission. And any time that chain involves a person who's immunized, that chain breaks and transmission doesn't happen. And so we can contrast this situation with one set up the same way, but where there are more people who are not immunized, or the fraction of not immunized people is higher. So that's this, where we've kind of we've decreased the number of yellow immunized people and increased the number of blue not immunized people. And we can just make more chains of transmission, and what we would see is a lot more infections. And so this is what happens when there isn't adequate immunity in the population. So this is what happens when we don't have herd immunity, and this is what happens, the previous slide, this is when we do have herd immunity. And this is what a vaccine attempts to do. It tries to drive up the population of people who are immune so that any little outbreaks that happen don't spread. And vaccines aren't perfect. They can't protect everyone. Some people can't take them, and other people, the vaccine just doesn't work quite with their immune system. So we kind of have to push to vaccinate as many people as we can. For some diseases and some pathogens, we know what percentage of people need to be vaccinated to prevent outbreaks. So we know that for something like measles, if 85% of the people in a town have been vaccinated, measles outbreaks won't happen, or big measles outbreaks won't happen. But if a lower percentage have been vaccinated, so only 80%, that could allow outbreaks of measles to happen. And so understand what we're saying here. If 80% of the people have been vaccinated, those people are not the ones going to get measles. We're thinking about the smaller group of people, will they be protected or not? And in this example, with 85% vaccinated, the other 15% are not going to experience an outbreak. But if this were lower, like 80%, the remaining 20% could experience an outbreak. So with vaccination, it's always a question of, can we get enough people vaccinated to stop outbreaks? That's the main thing vaccines try to do. The other thing they can do is lessen the severity of diseases, and that's primarily what we're getting from the COVID vaccines. I don't think we have much data yet on how they affect transmission, partly because we can't do those experiments yet, but also partly because we haven't vaccinated enough people to see what happens in a community of nearly enough vaccinated people. So this number, this 85%, would be more or less effective depending on how contagious the disease it is or how effective the vaccine is. And we could look at that. So we don't know the number for COVID. And I also want to mention regarding COVID that people who talk about herd immunity as what we should be looking for, they want us to intentionally infect each other with COVID to get herd immunity. Those people are delusional because they're only looking at COVID as a disease that doesn't kill everybody. What they see with COVID is that it only kills 1 to 2% of the people who get it. What they don't see is how many people get debilitating long-term syndromes post-COVID. And they don't see how many people spend several weeks unable to work with a very unpleasant virus. And we don't know things like how many people who get COVID are going to end up with chronic fatigue or paralysis or long-term lung damage, long-term heart damage, cognitive problems. All of those are things we've seen, and all of those are things that were ignored by the people who were talking about herd immunity as a intentional thing we should get. So if you're listening to people telling you what to do about COVID, make sure they know what they're talking about. And the last thing is, vaccine skeptics should know that what we're trying to do with vaccines is protect the people who are susceptible. So with any disease, there are groups that are more or less susceptible. And for COVID, it's seniors, but for a lot of other diseases, it's young children, and it's people who've been vaccinated and don't realize the vaccine didn't work in them. Because no vaccine gives protection to every single person who gets it. And so the people who aren't protected, they don't know it. And then lots of other people have compromised immune systems. So if they've had an organ transplant, or they have an advanced disease that disrupts their immunity, or they have conditions they're born with, or they're getting cancer chemotherapy, all these people are going to be more susceptible to any disease. And so as a society, we have to decide, are we willing to take vaccines to slow down infections and protect these people who can't be vaccinated? And I don't think there's anything I can say that will change the mind of a vaccine skeptic, because what we know is that people who embrace conspiracy theories, that's what they want to do. And not everyone who's a skeptic embraces conspiracy theories. And so I want to just real quick break down how a scientist sees this. People who have distrust of things like pharmaceuticals or the government, they have reasons for that. There are historical reasons why some people are more or less trusting of authority, and I can't argue against that. That's logical. People who believe conspiracy theories are not behaving logically. And I want to show you the test scientists can use to see whether that's true or not. And the test is, extraordinary claims require extraordinary evidence. So if someone is saying something crazy, like my neighbor is an alien, they have to have amazing evidence. They have to have dissected their neighbor and show that they're not human, or they have to have their DNA and show that it's not human, or they have to have something like that. they can't just say my neighbor acts weird, therefore they're an alien. And that's kind of the level of internet sleuthing that leads to conspiracy theories. What we see is, well, I don't trust companies. That's not an extraordinary claim. You don't need extraordinary evidence to say you don't trust companies, and I won't argue with that. But someone saying that billionaires created COVID, please. Not even a billionaire can gather enough technology to design a virus like that. Or somehow 5G signals, so electromagnetic waves, could somehow interact with nucleic acids. Spoiler, they can't. Those are bonkers. Those are incredible claims, people are saying. Or microchips, tracking chips in the vaccines. Well, people who claim to have found them are showing diagrams of other machines that they've just gotten off the internet. No one has found one of these chips. No one can replicate the supposed physics that make 5G start a virus. These are extraordinary claims that require someone showing exactly how it happens. They need documentation. And so that documentation has to be amazing in these cases. And internet sleuths don't come up with amazing documentation. That's just not something that has happened. So, when you're confronted with a new exciting theory, just ask yourself whether it's an extraordinary claim, and if so, does it have extraordinary evidence? And there's not much else I can say, and I know I can't change anybody's mind, but this is what I would like everyone to keep in the back of their minds. So thanks for listening. Again, you don't have to study anything that has these letters in the top right. Catch you later.